Happy Friday, everyone. This week I'm going to focus exclusively on a landmark set of studies published in Nature. They reported the results of the Roadmap Epigenetics Project. Well, what is this? It's the culmination of many years of work and over $300 million invested by the NIH. So in 2003, you can remember that the genome was mapped. That is the sequence of all the different amino acids that make up DNA. As we know, this has led to enormous accomplishments in autism genetics, but there was always something missing. There's another genetic code in our bodies. Think of DNA as a piano. You need someone to actually play the piano. The keys are DNA, but the way the piano is played is epigenetics. Epigenetics, or quote-unquote above the genome, literally taken, represent different ways in which our body can turn on or turn off expression of proteins from DNA. It isn't part of the DNA sequence, it's modification of DNA expression through different tags that turn on and turn off DNA translation into proteins. There's a few mechanisms by which this happens, and instead of explaining them, I really suggest you click on one of the links below the caption and look at the series of papers that was published. The site on Nature includes really great illustrations of the process. But for a more simplistic view, and something that I really thought was really fantastic, if you click the link to the PBS NewsHour piece, there's an amazing video that explains how epigenetics works. The announcement on Wednesday involved the publication of epigenomes in 100 different types of cells in the body. Previous work had used cell lines, which really aren't that helpful when you think about disorders that affect different organs of the body and different tissue types. Because epigenetic tags that turn on or turn off expression may differ in places like the brain, the blood, the liver, it was really important to go through these 100 different cells from the human body. Of really great importance for autism is that epigenetics is a mechanism by which early life exposures can affect disease outcome and how non-genetic exposures can affect gene expression. For example, we know that folic acid is protective against autism. This happens on a gene that is controlled epigenetically. We also know that things like diet and chemicals affect the genome. Now we can better understand how they relate to autism. Some important questions that can now be addressed in a way that couldn't before include things like, are epigenetics responsible for the difference in prevalence between males and females? We know that epigenetics is different in male brains and female brains during development, so this could be, but maybe isn't definitely, the answer. Another more theoretical question is, can we use data from blood the same way we can from brain? Are the epigenetic tags the same in blood and in brain? I think the answer is no, and I know we definitely need brain tissue, but now we can study this more. Researchers are already using this data to advance scientific discovery. For example, using the data from the Roadmap Epigenome Project, researchers were able to identify findings from their mouse model of autism, relate them to human tissue, and then identify a mechanism where changes in activity were reflected in the immune system. Unfortunately, when it comes to autism compared to genetics, there are very few studies looking at the epigenetics of autism. Last year at the International Society of Autism Research, Danny Fallon at Johns Hopkins School of Public Health led a session on environmental epigenetics and its role in autism. Epigenetics might explain why the concordance between identical twins is not 100%, and how certain environmental factors, as I mentioned earlier, like diet or chemicals, can increase risk. Epigenetics may be above the genome, but the markers can be heritable. This complicates the notion of heritability having to do with DNA sequence or specific genes. For example, a researcher in Canada named Art Petronas has an editorial that makes a comparison to height. We know that height is 80 to 90% heritable, but of the 40 areas of the genome that have been linked to height, only 5% account for the variation. It's not that height isn't heritable, and it's not that autism isn't heritable. It's just more than there are just gene mutations at play. We probably need to rethink the term heritability. Heritability doesn't just apply to the DNA sequence, and we need to know more about the epigenome to narrow down risk in ASD. In the environment, there are dozens of different environmental factors that affect epigenetic expression. We don't know all of them. This does make it very hard to narrow down a specific set of environmental factors. We have some ideas that diet and some chemicals affect expression epigenetically, but we don't know all of them. But even without a gene environment angle, the role of epigenetics is incredibly important for autism. Autism is a disorder of development in which the brain forms connections that shouldn't be there and are missing connections that should be there. Epigenetic modifications are important for development. When the egg and the sperm come together, it's epigenetic programming that helps the two fuse and then replicate to build an embryo. Finally, epigenetics signals regulate a number of genes already associated with autism, including FMR, those on a region of 15, renin, oxytocin, and a new gene associated with autism, CHD8, is involved in epigenetics as well. The publication of the Roadmap Epigenome Project makes it possible to understand the role of this mechanism in autism like it never has before. I really hope that in the coming months and years, we start to learn more about the epigenetics of autism, and if we do, it's because of the work of this Roadmap Epigenome Project. Thanks for listening this week. Please click the links below, those from the Nature article and the PBS NewsHour article. And also, please do not forget to register for the April 22nd Day of Learning Evening of Celebration. It sold out early last year, and we expect it to sell out early this year. Thank you, everyone, and talk to you next Friday.